Yeah, thank you very much for the kind introduction. I'm Leonie. I'm a second-year PhD student at the University of Cambridge, UK, and at the EBI, the European Bioinformatics Institute, and I'm part of the Pathogen Informatics and Modeling group. So yeah, my talk is about mechanistic modeling of Streptococcus pneumoniae, population dynamics after vaccine introduction. And I think I'd like to address the elephant in the room first. Why have a pathogen genomics talk at a workshop about the mathematics of the hallmarks of cancer? And I actually asked this question myself, and I came up with the following answer, which I'm not sure how well it translates, but so the idea is of this phylogenetic tree that with all the in-group of the talks about the mathematics of the hallmarks of cancer, I'm kind of the out-group that helps to group together all the rest and makes clear what kind of connections are between the other talks. But I hope that we're also going to get to the point where we can see some connections between at least my methods and maybe some concepts and ideas. But yeah, bear with me. I'm going to have to introduce a number of concepts and ideas just because I'm aware that probably most people in the room are not really familiar with pathogen genomics. Talking about this, Streptococcus pneumoniae is a bacterium. It's genetically very diverse. More than 100 serotypes are known so far. So serotypes are typically defined based on the proteins on the outside of the capsule. You might recall this from COVID, but unlike COVID, where usually serotype replacement was happening, the alpha wave and the beta and so on, in Streptococcus pneumoniae, they actually coexist in the same environment, and we know already more than 100. And Streptococcus pneumoniae typically does one of two things. So it usually has to colonize the host first, which is in our respiratory tracts, and it can live there harmlessly, but then it can turn pathogenic and cause a number of very severe diseases, including pneumonia and meningitis. And that happens especially often in young children and elderly people. And I don't have very current numbers on this, but in the year 2000, there were more than 14 million serious infections of Streptococcus pneumoniae and more than 800,000 deaths connected to this. So as per usual with bacteria, you can treat it with antibiotics, which I'm sure you're aware is problematic. We're running out of antibiotics we can use in general. And so the first line of defense is actually prevention, which you can do with pneumococcal vaccines. And so a number of those have been introduced in the last 25 years. The problem really is with the genetic diversity of Streptococcus pneumoniae, and because you usually target the serotypes, you can't possibly target all 100 serotypes at the same time. So instead, when you develop a vaccine, you have to go and select a number of serotypes that you want to target with the vaccine. And you usually do that by assessing how often a specific serotype turns pathogenic. And that actually differs quite a lot between the different serotypes. So when the first vaccine was developed, 7 serotypes were targeted in that vaccine. And what you expect from like a population genetics viewpoint to happen if you introduce a new selection pressure is that the frequency of those types will go down. So they will become less common of the vaccine types, and the non-vaccine types, so the ones that are not targeted by the vaccine, will actually increase in frequency. And this is also kind of what we see in the data. This is like a sketch of the data, but it's just to illustrate that in reality, it's a bit more complex than just everything decreasing what is a vaccine type and everything increasing and what is a non-vaccine type. And we're especially interested in the non-vaccine types and how much they increase, because it is definitely possible that among the ones that aren't targeted by the vaccine, some of them might cause disease quite a lot. We just don't know about it yet. So you really want to know what kind of impact the vaccine has on a population level. And so the two questions we're mainly interested in is which non-vaccine serotypes are the most successful post-vaccination, and then what gives them the selective advantage. And to answer those questions, we've developed a mathematical model But to explain the mechanism behind the mathematical model, I need to introduce some pathogen genomics ideas to you. So the first thing I want to talk about is bacterial pangenomes. When you look at 3 different genomes of pathogens, in this case, these in my example would all be Streptococcus pneumoniae sequences. You can define the pangenome, which is like the overlap of those genomes, and the bit in the middle that they all share is called the core pangenome, and then everything that might be shared between a number of them, but definitely not all of them, is called the accessory pangenome. And we're really talking about genes here. So those are the genes. The core gene pangenome is the genes that are shared by all of them, and the accessory genome are not. The genes are not present in all of them. And so you can see the diversity you usually have in bacteria is much higher. If we did a plot like this for humans, the overlap would be perfect because we all share the same genes. However, if we look at a pangenome of bacteria, this is Streptococcus pneumoniae, we see on the right-hand side this large peak for the core pangenome with the genes that they all share. And then here on the left-hand side, we have the genes that only occur once in this data set of over 600 sequences. And then the middle bit, we have the so-called intermediate frequency genes. So genes that occur in some of the sequences, but not in all of them. And this is really the interesting bit for us because it's a bit difficult to understand why that even exists from a population genetics evolutionary perspective. And I want to introduce the specific data set that I've been working with, or one of the data sets I've been working with, which is from Massachusetts in the early 2000s. And they collected nasal swabs from children before and after vaccine introduction. And basically in this plot, you can see in orange the vaccine type sequences and their frequencies are correlated between pre-vaccine and post-vaccine. So you can see that they all go down in frequency. And then in blue, you can see non-vaccine types that increase in frequency. In black, we have a number of mixed clusters, and they show quite a good correlation, but in general, I think you can tell, even though there is some correlation between the pre and post-vaccine time points, you can see that there's quite a bit of change going on. However, if instead we look at the pangenome, and now we're specifically looking at the intermediate frequency genes, we can see this striking correlation pre and post-vaccine. And so this is actually quite surprising because you would think that if there's so much replacement going on, usually then the genes should change as well, right? Especially in the intermediate frequency genes, it seems likely that then there would be quite a lot of variation going on. And so in my model, we're trying to use a mechanistic explanation as to why this doesn't vary so much. And there are a number of possible explanations. And one is the adaptation to different niches, which is like a general concept in evolution. If you have different niches that are occupied by the same species, they usually tend to develop independently. And so an impact, like the vaccine might not have an impact on the pangenome of a separate part of the Streptococcus pneumoniae population. But this is quite unlikely to be true for Streptococcus pneumoniae, since its only niche is the human respiratory tract, and there's not so many niches to occupy there. Then secondly, it might be due to random genetic drift, and for sure random genetic drift plays a role in the system, but it's quite unlikely to cause this high correlation. And so another explanation would be negative frequency dependent selection. And so this is another concept that I would like to introduce because it plays a major role in my model. So the idea of negative frequency dependent selection is based on selection. I'm sure you all know about negative selection, where if something doesn't provide any benefit to the organism that carries that specific gene or SNP or allele, it will eventually in evolutionary time go down in frequency within the population and die out eventually. And similarly, for positive selection, if something's beneficial, you would expect that over evolutionary time trajectory, that gene eventually will be present in every single organism in the population. Negative frequency dependent selection has that idea that there are genes that have an ideal frequency that is neither 0 or 1, that's somewhere in between, and that it is selected for if the frequency is below that ideal frequency, and it's selected against if it's above that specific frequency. And so this is a concept that is actually quite well established nowadays in population genetics for bacteria. But I'm aware that there are basically one of two possible reactions to this concept. Some people find it very intuitive and other people find it very questionable. And so I would like to introduce a couple of examples where NFDS really seems to make a lot of sense in population genetics for bacteria, but I'm really happy to discuss further after my talk. So one example for negative frequency dependent selection in bacteria is bacteriocins, which are toxins that bacteria use to kill each other off, basically to have a selective advantage. And so in this example, this orange Streptococcus pneumoniae produces this bacteriocin with which it can kill off this green bacterium. And it also has an immunity protein, which makes sure that it doesn't kill itself by mistake. And then there is a third group of Streptococcus pneumoniae here, who are called the cheaters, because they only have the immunity protein, but don't waste any energy on producing the bacteriocin. And now if you simulate this over time in a very simple manner, you would expect that eventually on a population level, these will reach equilibrium and all of them will be present at 1/3. Or that might depend on the fitness cost and fitness levels that these three actually have. But this is kind of one of the examples for which negative frequency dependent selection makes a lot of sense. Another good example is the uptake of specific sugars. If there is a bacterium that can take up a sugar that none of the other bacteria can take up, it provides a strong advantage for that bacterium. But once everyone has that gene, everyone can take up that sugar, actually there won't be that much sugar left in the environment and the benefit will actually decrease. Okay, now I've finished to introduce all the different ideas and concepts that lead up to the model that I've been working with. It's a stochastic Wright-Fisher model, so it's a Markov chain, and it's based on a previous publication by Karanda et al. And so it's my model. So the model that I've been working based off is an individual-based model. My model is actually a compartmental model. So the way that works is we cluster the sequences by genome similarity together into different compartments. And then we can simulate, for example, forward in time for the model. In this case, we have 4 different compartments, and you can see the frequency changes over time. And my model has 5 free parameters. And there's a migration term that just makes sure that we allow migration of new bacteria into the system, which is essential in this well-connected world that we're living in, to allow other, basically humans, to bring in new Streptococcus pneumoniae. Then the vaccination term, which is essential because that's kind of the effect we want to study in the population. And then we have the negative frequency dependent selection, which actually has 3 parameters in total. And I will talk about this in a bit more detail. And I don't want to spend too much time with the equations, and I apologize for the colors. I usually give this talk in front of bioinformaticians. In general, we have like the counts, which is the size of the different compartments. And we draw from a binomial distribution to get the migration counts into the system, which are distributed equally between the different compartments. And then we draw from a Poisson distribution that depends on the fitness of the different compartments or strains, as I call them here. And so essentially, we're checking whether or not a compartment is a vaccine compartment or a non-vaccine compartment, so whether it's negatively affected by the vaccine. Then we have an influence of the migration term, and then we have the whole NFDS term, which essentially penalizes genomes that have genes that are already over the equilibrium frequency, and encourages genomes or sequences to grow, types to grow, that have a lot of genes that are currently below that ideal frequency. And so what we can then do with this model is to fit it to data. As I said, one of the data sets I've been working with is from Massachusetts, from those nasal swabs. And basically what we get is we have 56 clusters of the genomes, and we take the data from the first data point in 2001 directly from the data and put them into the model as the start value. And then we have 2 consecutive data points, and we fit the model to those data points. And this is what the fits currently look like. It's a lot to take in. I acknowledge that. In general, we are quite happy with the overall fit. And I'd like to discuss a bit more in detail the kind of quantitative results that we did. So one nice thing about Bayesian modeling is that we can do really nice model comparison. So something I've done is to check whether this 5-parameter model that I essentially based off the previously published model, whether that is actually the best model that we have. So I checked whether the NFDS is well parameterized with the three parameters that it currently has. So those parameters are a strong selection term and a proportion of the intermediate frequency genes that are actually under the strong selection, and then another selection term that is weaker and that applies to all other intermediate frequency genes. Now, I compared this against a null model that doesn't have any negative frequency dependent selection, and I compared it to a 3-parameter model that has just the same NFDS strength across all intermediate frequency genes. And I also compared it against a 4-parameter model that has a strong NFDS term and the proportion, but it doesn't have any weak NFDS. So that's set to 0 by default. And so because we're doing Bayesian modeling, we can do a likelihood ratio test for this, and turns out that actually the 4-parameter model is the best model we have. So basically, it turns out that this weak selection pressure doesn't really have any, there's nothing in favor for this parameter, so it's better to drop it. And we actually the fit that I showed you on the slide before was with that 4-parameter model already. And so we can, for example, take a look at the parameter values that we actually get from the Markov chain Monte Carlo fitting. And in this case, for example, for this specific fit for this specific data set, we get that 30% of the NFDS, of the intermediate frequency genes are under this NFDS term. Right. And questions we'd like to answer with this model is which genes specifically provide the selective advantage for the most successful non-vaccine types, and is that consistent across different data sets? So I'm at the moment doing quite a bit of bioinformatics data wrangling to make sure that I can compare different Streptococcus pneumoniae data sets from around the world. And then an important public health kind of question, which additional serotypes should be included in future vaccines. Now, as I promised, I am going to quickly talk about what does this have to do with cancer? Does this have to do anything with cancer? Maybe you're all fit to answer that better than I am. I just came across very recently one of the first, I believe, negative frequency dependent selection papers in cancer, and I thought it was a really interesting read. I'm aware that this might be quite a controversial topic, especially for people who prefer like very parsimonious kind of models. But I'd be really keen to hear what people think about this. And yeah, with this, I'd like to thank my team, especially my supervisor, John Lees, and all of you for your attention. Thank you.